So many of the organisms that we'll be studying this semester are indeed prokaryotic, but as you're aware, some of them are eukaryotic. And, naturally, there are some differences between prokaryotic and eukaryotic transcription and translation. Although they're pretty similar, they're different in a number of different ways. The start codon is also AUG, but it codes for a slightly different form of methionine. You'll recall prokaryotic messenger RNAs often contain information for several related genes. Eukaryotic messenger RNAs code for just one protein. Also, eukaryotes have a nucleus, and the DNA is separated in the nuclear compartment. That means that eukaryotic transcription and translation cannot be simultaneous because the transcript has to leave through the nuclear pores in the nuclear membrane and be carried to the ribosomes, which are located in the cytoplasm. It's in the cytoplasm, then, that eukaryotic messenger RNA will be translated into proteins. In addition, most eukaryotic genes do not exist in an uninterrupted series of triplets that code for a protein. There are interspersed sequences that do not code for the protein. These interspersed sequences are called introns. The coding regions, in contrast, are called exons. As you can see here in this DNA template, there are expressed pieces, or exons, and then introns, interspersed pieces. When that DNA template is transcribed into the primary messenger RNA, the first transcript, we'll see that there are exons and introns all the way along that piece also. These so-called split genes require further processing before they're translated to remove these interspersed sequences. That's the process of RNA splicing. In RNA splicing, a lariat forms, or a loop, in the interspersed sequence, and a protein and RNA complex called a spliceosome, this blue guy here, comes along and literally snips out the intron at its ends and sticks the two exons together. So now we have just one piece of fully coding, mature messenger RNA, and it's ready to leave the nucleus. This is another place where we see evidence that archaeans are more closely related to eukaryotes because they also have split genes. Now it's interesting that some of these introns do actually code for substances. In humans, introns seem to represent about 98% of the DNA. Although we used to not understand their relevance, the discovery of the extreme importance of this intron material has really revolutionized genetics, because these introns actually allow creation of new proteins from the same gene. We used to think that there was one gene to one polypeptide. However, we now understand that one RNA transcript could have several possible options, even hundreds of alternative messenger RNAs that could be made through them, through the process of alternative splicing. As you can see over here, we have a pre-messenger RNA with 4 different genes in it. Through alternative splicing, we could end up with this mature messenger RNA or this mature messenger RNA, each of which would code for a slightly different protein isoform or different version of that protein. This discovery means that we now understand that in a eukaryotic genome, we can actually produce more polypeptides than there are genes. In fact, we've discovered that in 40% of human genes, there's some form of alternative splicing, to the extent that there could be hundreds of alternative messenger RNAs created from one gene. Another process that we're starting to understand is RNA editing. This involves alteration of the sequence of nucleotides in the primary RNA transcript before it's translated. This produces a messenger RNA that's actually not even truly encoded in the genome. So this is some fascinating stuff on the forefront of genetics at the moment. Unfortunately, we don't get to spend too much time in it in this course. Let's take a quick look at an animation of RNA splicing so you can get a better understanding of that process.
RNA splicing removes introns from pre-mRNA and connects the exons together. Splicing involves a complex called the spliceosome that has subunits called SNRPs. Each SNRP contains a small nuclear RNA and proteins. Certain sequences are required in an intron for it to be removed by the spliceosome. Among the requirements are a GU at the 5' end of the intron, also called the 5' splice site, and AG at the 3' end, or 3' splice site. A branch site toward the middle of the intron is also required. This sequence contains an adenine that plays a key role in intron removal. The first step is binding of the U1 SNRP to the 5' splice site, followed by binding of the U2 SNRP to the branch site. Next, the trimer of U4, U6, and U5 SNRPs binds at the intron region, completing the spliceosome assembly. The 5' splice site is cut, and the 5' end of the intron is connected to the adenine in the branch site to form a structure called a lariat. At this time, the U1 and U4 SNRPs are released, and the U6 and U5 SNRPs shift positions. Then the 3' splice site is cut and the exons are connected together. The lariat intron is released along with the remaining parts of the spliceosome. The spliceosome subunits will later dissociate from the lariat, and the lariat will be degraded. The final outcome is that two exons have been covalently connected to each other, and the intervening intron has been removed.
Again, this is more detailed than you need, but hopefully some motion helps you understand the process. Previously, we've covered the genetic nature of viruses. They essentially consist of one or more pieces of DNA or RNA enclosed in a protective coating. But above all, they are genetic parasites that require access to their host cell's genetic and metabolic machinery to be replicated, transcribed, and translated. The genetics of viruses are quite diverse. In many viruses, the nucleic acid is linear. In others, it's circular. Most of them exist in a single molecule. However, in a few instances, there are several smaller pieces of DNA. Many contain double-stranded DNA or single-stranded RNA, but as we've explored previously, other patterns do exist. A couple of things that are consistent for all viruses, though, is that the viral nucleic acid penetrates the cell. The nucleic acid is introduced into the host's gene processing machinery. That virus then instructs the host machinery to synthesize large numbers of new virus particles, and the viral messenger RNA is translated into viral proteins on the host cell ribosomes using host's transfer RNA. So hopefully this puts all the pieces together for you. Of what machinery animal viruses are really hijacking during their infectious cycle. Before we go on, let's have a quick review of the different means of processing gene information in prokaryotes versus eukaryotes.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.